Today, I would like to throw the light on the natural products because natural products are part of our life, and I would like to emphasize on different kinds of natural products, how they have been important part of in our life, and how they have contributed to our well-being. Yeah, before going to my talk, I would like to give emphasis about our institute, where IICT comes under CSIR network, which is the part of the Ministry of Science and Technology, Government of India. CSIR has 37 laboratories of different disciplines like biological sciences, chemical sciences, engineering sciences, information sciences, and physical sciences. So this Council of Scientific and Industrial Research has established in September 1942 as an autonomous body under the Ministry of Science and Technology. And almost all over India, there are 37 laboratories and other some extension units of main laboratories also. In Hyderabad, we have three laboratories: IACT, CCMB, and NGRI. IACT comes under Chemical Sciences Cluster. NGRI comes under Physical Sciences Cluster. CCMB comes under Biological Sciences Cluster. So then coming to IACT, IACT has been established before India's independence in 1944 by erstwhile Nizams in the name of CLSIR, Central Laboratories for Scientific and Industrial Research. And after India's independence, it has been taken by Government of India and then renamed as a Regional Research Lab, Hyderabad. And in 1989, based on the technology developed by under the leadership of Honorable Dr. A.V. Ramarao, the name has been changed to Indian Institute of Chemical Technology in again in 2002, 12. Since it comes under the network of CSIR name has been added before the name of IICT. So since then, almost from the last almost 8 decades, IICT is contributing to the society in the form of drug development and developing the process for active pharmaceutical ingredients and different sectors. So this is the main divisions of IICT. IICT main strength is chemistry and process chemistry and chemical engineering. We have different divisions including catalysis, where we develop the heterogeneous and homogeneous catalyst for the different reactions, and a genetic chemistry laboratory, diversity oriented synthesis lab, fire chemicals lab. And see, these three labs majorly focused on the process development and total synthesis of drug intermediates, and agrochemicals and fluoroorganics lab, which is mainly focused on development of the agrochemicals and fluoroorganics lab. And this is the only lab which is working on the fluorination of different organic compounds. And natural products laboratory, where I am working in this lab, mainly focused on the isolation and synthesis of different kinds of natural products derived from different sources of natural products. And in addition to this, it is always supported by the different analytical centers like NMR, MAS, and XRD, and other polymers, material, molecular modeling, different divisions also. We also have the state-of-art animal house facility for the drug discovery purposes and other purposes. So when coming to today's topic, importance of the natural products, as we are facing different seasonal diseases and other diseases. Particularly, this is all because of the imbalance in the atmosphere, which causes or incidence of different diseases. You see now in south there are very less rains, but in the north there are huge floods. Similarly, unusual temperatures, unusual rains, and this is all because of the imbalance in the atmosphere. And also at the same time, we are also removing all the forest plants deforestation, and which leads to the global warming and finally leads to the incidence of different diseases. And also another cause is increasing the drug resistance. I think many of us might have been utilized or consumed the antibiotics, particularly in the COVID pandemic period, but particularly antibiotic like azithromycin, we must have taken several doses. Now, even if you have cough, now 5 tablets is not enough. It is because of there is a resistance build up in our body. So this is also one of the major cause for our healthcare. So these are the major diseases in the world. Here I have listed some few: AIDS, tuberculosis, diarrheal diseases, malaria, hepatitis B virus, avian flu, tobacco related disease. So here you can see for every disease almost more than million, at least 1 million deaths are happening every year, and more than 1 million deaths are for new cases are finding. So in certain diseases there are no globally available treatments for the diseases, particularly viral diseases and AIDS and tuberculosis. If it is there in the critical stage, even though we have also experienced the COVID pandemic where uh, there are no global treatments, even though you are rich enough. That is a major drawback. And some of the lifestyles, our lifestyle is also contributing the bad health of ours. Here you can just you can see home theaters, multiplex in the living room, and the whole family here they engaged by in the cell phones, and this guy is uh, busy with this burger. So the modern lifestyle habits also contributing bad health of the human beings, and that causes to obesity. That is a major problem in the present society. Even we can observe the obesity kind of things in the children's also nowadays. This is all because of the our lifestyle food habits. So if you see, think about the drug, the always getting a drug to market cost about the $1 billion, which is a high costly drug, and it of course it's expensive. And of course, even though it is expensive, there are no drugs available for the critical global diseases. And if you can use the synthetic drug, that may not be sustainable long term, because that is also having their own side effects, as well as the other effects also, resistance problem. And in that case, in that aspects, can we think about the traditional medicine, that is plant based medicines, where if you see, there are almost 50 to 60% of the present marketed drugs are originated from the natural products. So the only issue is the very poor quality control. But in the present scenario, having the revolutionary techniques in the analysis, biological revolution, we can come across for the quality control aspects of the herbal drugs. What is a traditional medicine? Because traditional medicine is, it's a medicinal knowledge and innovations and practices of the indigenous local communities around the world. And they do not have any like a medical degree, but they got this the experience, the knowledge from their experience. And this experience has developed gained over the centuries. If you see, in the ancient times, people don't have did not have that much knowledge, but based on their knowledge, they have identified the medicinal plants, surrounded them as a medicinal plants and toxic plants. So that knowledge has been rooted to the generations together, and then formed as it is as a traditional medicine. So as upon, we can call it as a like a Indian system of medicine, Chinese system of medicine. That's what every country has its own system of medicine. That is the main reason for formation of those medicine, traditional medicine. Of course, in India, traditional medicine can be classified two categories. One is codified system and a non-codified system. Codified comes under Ayurveda, Unani, Siddha, Homeopathy, Ayush Ministry, and non-codified folklore system. This folklore system, majority people, the tribal people will use this folklore system, and even though they don't have any scientific evidence, but uh, the lot many population are using in the tribal areas or interior forest of the every corner of the country. Of course, both the systems are contributing equally to the primary health care in India. So the of course traditional medicine is also complementing the Western medicine, which is also called as a complementary and alternative medicine, and also interchanging the traditional medicine in some countries, but raw material is the same, which is a medicinal plants. Of course, by the different name. In US it is called botanical drugs. In India it is called herbal drugs. In Europe, though, that is called nutraceuticals. So traditional medicine is serving the mankind, I think, for the from the last 4,000 or more years ago. But in the present scenario, we can do much better. And this is in the developed countries also. It is accessible as over to the counters, OTC counters. And here just I gave some of the figures where in Iran, Colombia, South Africa, and China. And these type of people who can come across when you visit the tribal areas, still their entire life they are dependent upon the plant-based medicine like a folklore medicine, and even though they do not have any like RMP, PMP, whatever the degree, but they have they got this knowledge from their fathers and forefathers, and they are serving very well in the tribal areas. Particularly when you see the tribal people, India is the largest one of the country having the largest population of tribes. Of course, this is a heterogeneous tribes and distributed in the entire corner of the country: Himalayan region, northeast region, south region, western India, Andaman and Nicobar Islands. So based on this, recently World Health Organization also conducted a survey regarding the plant-based medicine, which revealed that in the globally more than 80% of the people, world population are relied on the plant-based medicine, traditional medicine for their primary healthcare needs. And further, it has also listed another 21,000 medicinal plants uses around the world. And when coming to India, we have 16 distinct agroclimatic zones, and majority of the almost 45,000 different plants are well utilized in the our systems of medicine, traditional system of medicine like Ayurveda, Unani, and other system of medicine. And some of the medicinal plants which are we are also fairly exporting to other countries also. And currently another 5,000 products are included in the pharmacy of Ayurvedic treatments. So these are the major natural sources like plant kingdom, microbial world, animal sources, marine world, and venoms and toxins. Among this, plant kingdom has served a lot to the human being in the form of Ayurvedic medicine and other medicines. Like microbial world, penicillin is the one of the best example where it has been discovered from the microbial world. If you see marine world and animal sources, in the present market, there are a lot of antibiotics and other drugs which are has been discovered from the marine world and also animal sources, and some of the drugs also discovered animal venoms and toxins. If you see the different applications of natural products, so as we have seen that the discovery of artemisinin, which is the best drugs till date for the combat the anti-malarial. So the artemisinin has been for the discovery of artemisinin, the Chinese professor also rewarded Nobel Prize in medicine. So that is one of the best example in the application of natural products. And also in the other fields also natural products are used in a very high manner, like ivermectin B as a crop protectant, rosmarinic acid food preservative, and trapaxin B biological source, and epigenin very well utilized in the beverages industry as a colorant, and linolol as a perfume, and stevioside as a artificial sweetener derived from stevia rodendiana, and linoleic acid as a lotion in very highly utilized in the perfumery industry. So, and if you see here the beauty of the nature. So just the one plus isomer gives orange smell, plus limonin and minus limonin gives a lemon smell. Similarly, the spearmint cells minus carbon and plus carbon. Likewise, there are many this type of compounds in the nature where plus isomer is a drug and minus isomer is a toxic compound. So if you see the vaccine adjuvants, so we must have familiar with this compound like QS21, which has been isolated from the Mexican American border plant Collaja saponaria. This is a part of many vaccines, including Covishield vaccine. This is the recent application of the natural products. Particularly, this is the this QS21 is adjuvant, which we potentiate the immunity response. That's what this is the major. This is a part of major vaccines, including Covishield. This is a best one of the best recent example. And likewise, this, several other only source is natural product. There is no synthetic source so far. Likewise, there are several other drugs also. The only source is natural product so far. No synthetic process also been developed. Is a critical. And recently, particularly in the COVID and one and two period, two universities from Maharashtra, they have conducted a survey regarding the consumption of spices, because spices is a part of our tradition and part of our life. Even in our daily life, we consume lot of spices, including the curcumin and curcumin and mirialu, and then ginger, garlic, tamarind, and chili, lavanga, dalchini. So there are, we consume a lot of spices as part of our tradition and as part of our lifestyle. So they have conducted a survey regarding the consumption of spices versus viral load. So they have conducted starting from Kashmir to Kanyakumari, including other union territories also. Finally, they came to know that after the survey, that Lakshadweep people has consumed spices like anything. They consumed spices a lot. So that's what, there is no even single death has been recorded in Lakshadweep island in the particularly in the period of COVID. So these results also show eye opening, like the all the spices or whatever the spices we consume contains the polyphenolic compounds like anthocyanins and other type of compounds which has the properties of adjuvant property, that is which will increase the immunity response. So this is the one of the recent thing that majority of the scientists now focusing on working all these our regular spices to isolate and characterize the adjuvant properties of compounds, adjuvant properties of compound to combat the different vaccines. Like natural products has a long history of their usage, which has been traced back to our Mesopotamian civilization 2600 BC. Similarly, in the Egyptian civilization 1500 BC, there are almost 700 drugs were documented at that time. And when coming to famous Chinese system of medicine, which has been traced back to 1100 BC, in that period, almost several drugs has also been documented. And these natural products are traditionally established in the form of the system of medicine. Every country has their own system of medicine. Of course, in our country, Ayurveda is which is a long established system of traditional medicine practiced in India thousands of years. Now it is getting worldwide attention as an alternative system of medicine. Of course, even though Ayurveda speaks about like a concentration, extracts and fractions, but based on the Ayurvedic principles, several drug leads were identified and invented, which are in the present market. Of course, as a modern medicine. Here, I just quoted few like Ravulpi alkaloids to reduce hypertension, soralins, olarihenna alkaloids to combat amoebiasis. Similarly, L-dopa and piperidins, source of piper species, and picrosides, Picrosia curora, phyllanthus, antivirals, very well known, and withanolides, steroidal lactones, and glycosides, known as immunomodulators. And these are all economically affordable, and of course, easy procure raw material also very easily we can procure, and of course, the toxicity also less when compared to the modern medicine. And these are very effective, particularly in the chronic diseases. And coming to toxicity, these compounds, these fractions has to be consumed as per the dosage or which is advised by the Ayurvedic physician. Then only those are effective. Otherwise, if the dosage exceeds, the consequences will be in the organs, particularly in the kidneys. That is a major drawback of herbal medicines. So, if you see the recent winning Nobel Prize winning concepts, almost these concepts also related to our traditional system of medicine, particularly in the 2018 Nobel Prize in Physiology for their discovery of cancer therapy by inhibition of negative immune regulation, indirectly related to our Vata Pitta Kapha Ayurvedic concept. And in 2016 for the discovery of mechanism of autophagy, which is almost related to our fasting, which is called in Telugu it is called Lankanalu. So this shows the importance of our system of medicine, and emphasizing the importance of our system. When you see the biodiversity strength, almost among the 34 mega diversity hotspot globally, our country India hosts three hotspot. One is Indo-Tibetan region, Himalayan region, and Western Ghats. In these three regions, there are a lot of medicinal plants which are not even identified taxonomically, but the tribal people in these three regions are utilizing very well for their healthcare, and some of their plants also they are fairly exporting to other countries. In those areas, these tribal people who are working or advocating this folklore medicine, those are called traditional healers. And of course, in the recently, Government of India also conducted a survey regarding the existence of traditional healers. Almost to 3 to 5 lakh people who are called traditional healers are existing in the country, and fairly they are serving to the neighboring people based on this folklore medicine. And when coming to India's strength in herbal technology, fairly a majority of the plants, almost to 325 plants, are using as a pesticides, and another 425 plants are using as a gums, resins, and dyes, and some are in fibers, and some are in edible plants. And the contribution of India's strength in herbal technology, like Indian system of medicine, almost 1,000, close to 1,000 species are well utilized in the Ayurvedic system of medicine, and more than 700 species are in the Unani system of medicine, and more than 600 species are Siddha medicine, 250 to 300 species are in the Amchi based medicine, that is a Tibetan based medicine, and even of course 30 to 40 species are also well utilized in the modern medicine. Now in the recent trends, the after the patient who are undergone to the chemotherapy of cancer patients, now doctors are prescribing plant-based nutraceuticals instead of advising the tablets and all those. This is the recent trend in the particularly herbal-based nutraceuticals in the patients. When you see the drug discovery, number of drugs, natural product-based drugs discovered from the last 4 decades, almost close to 2,000 marketed compounds derived from natural products were marketed, and from 1981 to 2009, even though still another 90% of this gold mine is as it is, and only 10% flora has been explored till date in India, as India is called as a botanical garden of the world. When coming to the fungi only less than 1% has been studied. So this shows a lot of opportunities in the area of natural product to explore the active compounds to combat the different diseases. When coming to the contribution, so natural products has a lot of contribution, particularly in the antibacterial drugs, anti-cancer drugs, and anti-hypertensive drugs. So still amphotericin B, which is called as a gold standard, and lipopeptide caspofungin are derived from natural products. Similarly, particularly in the dengue virus, in the dengue, the papaya leaves, which are the part of the Siddha drug, is the only source which is known as a platelet aggregator for the dengue, to combat the dengue. Similarly, there are other drugs, several drugs which are having a lot of importance, which are discovered or identified from the natural products. There is no synthetic source so far. So here I just gave some of the best selling drugs worldwide which are originated from the natural products. So based on the quality control aspects, plant-based drugs has been divided into 4 categories. Category 1, which comes under folklore medicine, where there is no scientific sanctity for these herbals. And category 2 comes under the Ayurveda and Siddha, other drugs which are documented in herbal medicines in the treatises and books. Category 3, modified herbal medicines, polyherbal formulations, some amount of the safety and efficacy data required. Category 4 is completely imported drugs, where the both countries should be registered, and also complete safety efficacy data should be required. So then only those medicines will fall under the category 4. When you see the important herbal drugs in the developed countries also, more than 50% of people are using the plant-based medicine in, of course, developed countries and developing countries also. Of course, they, as I told you, maybe name is different. In US, it is called botanical drugs. In India, it is called herbal drugs. Similarly, in the Europe, those are called nutraceuticals. So when natural products, why these natural products are important? Because the natural products contains primary metabolites and secondary metabolites, and the primary metabolites are itself useful for the organism or plant for their growth, and the secondary metabolites are the main compounds are which are generated from the by the defense mechanism pathway. So these are the main real drug candidates, and there are different types of secondary metabolites like nitrogen containing compounds, polyphenols, and terpenes. And nitrogen containing compounds are called alkaloids: nicotine, morphine, cocaine, caffeine, glucosinolates are best examples. And phenolic compounds, flavonoids, anthocyanins, salicylic acid, lignans, tannic acid, allergic acid, and terpenes, limonoids, azadirachtin, saponins, and pionin, camphor. These are all the examples for the second metabolites. When you see the evolutionary techniques in natural product chemistry, in the early 1800th century, 1800, 18th century, there are no technology available in that case. In that time, the chemists and pharmacists are used to characterize those compounds based on the physical characteristics and chemical characteristics and chemical transformations. But keep on coming, moving to 19th century, some spectroscopic techniques are available like HPLC and UV visible IR spectrometry. But in the present scenario, even even 3D NMR technique is also available for the characterization of the complex natural products, including even X-ray technique is also available. So these are the evolutionary techniques in the natural products. And in the success history, particularly in the before 1800s, the people are used to take the natural product extracts, decoctions, concoctions, or crude fractions for to combat the different diseases. But in 1803, the the morphine was discovered from the one discovered from the plant. And this is the one of the best example where the traditional medicine, from the traditional medicine, people has discovered the first compound morphine as a drug candidate, which has further become a drug. So by following this discovery of morphine, so several other drugs also, compounds also discovered from the different sources of natural products, and majority of them are became a drugs. And when coming to semi-synthetic drugs, aspirin is the first semi-synthetic drug which has been marketed by the Bayer in 1899. And coming to the penicillin, penicillin has discovered from the Penicillium notatum in 1929. After that, in the period 1940 to 1970, in the 30 years period, majority of the antibiotics like tetracyclines, streptomycins, and doxycyclines and lipopeptides has been discovered. That's what this 30 years period is called golden era of antibiotics. And when coming to marine investigation, 1970s onwards, the intensive investigation now has been began. And after that, majority of the anti-cancer drugs and antibiotics has been discovered from the marine sources like marine sponges and marine fauna flora. So this is the drug discovery from natural products. There are different steps involved, starting from the raw material to drug candidate. So this is a lengthy process. Of course, it's a too costly process also. If you see the starting material, first step in the bio raw material. So raw material may be different natural products like from plants, fungi, insects, and marines and microbes. So that will be processes to prepare a bioactive extract. So for taking the extraction, there are different techniques available, starting from oxalate extraction, counter current extraction, and ionic liquids, pressurized extraction, solid phase, supercritical carbon dioxide extraction. So based on the compound nature, the extraction techniques will be adapted. Once the extract has been prepared, then that has to be processed by by using different chromatography techniques to get the pure compound. Here, chromatography techniques also, there are a lot of techniques are available, starting from the gradient chromatography like and, preparative TLC, column chromatography, and HPLC, UHPLC, counter current chromatography, supercritical carbon chromatography, membrane filtration, reverse phase chromatography, Cephardex, and there are different techniques available. But based on the compound nature, these techniques will be adapted for the purification of the compounds from the crude extract. Once purified compound is in hand, then structural distillation should be done. So there are different techniques, spectroscopic techniques, starting from UV to X-ray available. So every technique has its own significance. UV spectra gives the information about the chromophores available in the compound. Similarly, visible FTIR spectra will give the information about the functional groups present in the compound, and NMR spectra will give the number of carbons, number of hydrogens, and carbon-carbon linkage, carbon-hydrogen linkage, and mass spectra will give information about the molecular weight of the compounds, including fragmentation pattern, and X-ray is the ultimate technique to give the complete structure of the compound. once the compound is characterized, that has to be prepared in the large scale for extensive biological studies, and here the medicinal chemistry approach will come into the picture to prepare the different compound library for enhancement of the activity and reduce the toxicity. And once it is done, then it will be processed to pass the different phase 1, phase 2, phase different phases to become a drug candidate. So these are the three steps involved, majority most important three steps involved in the natural product drug discovery: identifying the hits to lead and lead optimization technique. So natural products activity is a multidisciplinary activity, as it has to be validated scientifically based on the uh, Ayurveda, folklore healers, and ethnobotany. Finally, it will be supported by the chemist, and finally it is validated by different biological techniques like cancer biology, neurobiology, and biochemistry. So there are different thrust areas available in the traditional medicine, starting from the standardization, quantification, and phytochemical, pharmacological screening, metabolomic study, DNA barcoding, herbal therapeutics, pharmacokinetics, marker profiling, phytoequivalence, phytoinformatics, and reverse pharmacology, high throughput screening, safety evaluation, value added drug delivery system, quality control and standardization, globalization of the. There are different traditional thrust areas, research areas in the traditional medicine, and just I would like to give some of the extraction techniques available in the natural products chemistry. And there are different techniques available, starting from the soaking, hydrodistillation, oxalate extraction, maceration, supercritical extraction, and refluxing. And when coming to spectroscopic techniques, and as I told you, NMR is the main spectroscopic technique which gives information about the number of protons and number of carbons, and also their linkage, carbon-carbon linkage, carbon-proton linkage, and COSY, and today NMR, depth, NOSY, and HMBC, HSQC, and other spectroscopic techniques, GCMS, mass spectrometry, where mass spectrometry will give the information about the molecular weight of the compound, and there are different ionization techniques are available in the mass spectrometry, and based on the compound nature, non-polar and polaric nature, the ionization techniques will be adopted. Electron impact ionization, chemical ionization, electrospray ionization, and fab mass, fast atomic bombardment, and MALDI-TOF. So high molecular weight compounds, MALDI-TOF will be adopted. And main approaches, there are different approaches in the natural products. Like first one is bioassay guided isolation procedure, and reverse bioassay guided isolation procedure, that is also called random approach. And NMR based or mass based approach, this is also called dereplication technique. And metabolomic based approach, biochemometrics, and molecular networking based approach, where this is also called almost related to the artificial intelligence. So the technology drivers in the natural world, as I told you, this traditional knowledge, folklore medicine are the main backbones of natural food chemistry. Finally, extracts has prepared and high throughput screening will be done, and the dereplication techniques, a lot of techniques, hyphenate techniques are available to know or pinpoint the known compounds, or to eliminate the known compounds and identify the new compounds, and purification of the structures and optimization development. Now 3D NMR technique is also available for the determination of the complex natural products. And just I would like to give some of the famous drugs which are originated from different natural sources. Here drugs from terrestrial plants. Artemisinin, which is anti-malarial drug. As I told you, for the discovery of artemisinin has been rewarded the Nobel Prize to the Chinese professor, and in 2015 this was artemisinin was isolated in 1972 from the Chinese medicinal plant Artemisia innova. And this is also as a small background story, when Vietnam in the Vietnam War, Vietnam soldiers were died because of the mosquito bite, not by the attack of the opposition soldiers. So in that case, the Vietnam has approached to China to help in that case. So China has at the time China has put up a high power committee under the chairmanship of the lady Professor Yo-Yo, and they have screened almost 15 to 2000 medicinal plants which are very well known in the traditional system of medicine for the malaria. And then finally they ended up with Artemisia innova by using different bioassayer approach. They got the activity in the artemisinin ANOVA fraction. So based on that activity, they have isolated artemisinin. Finally, that has become a drug. Artemisinin basically structurally it is a highly oxygenated sesquiterpene lactone containing a peroxide linkage. Peroxide linkage is very important for the activity. So this compound is very active against the chloroquine resistant malaria parasite, and it kills parasite during the early stage of its development, which is responsible for unprecedented success of this compound. So based on the artemisinin discovery, several other two derivatives also prepared and developed by one derivative artiflin, where they have been added chloro CF3 group into the artemisinin, and the another two derivatives, methyl derivative and ethyl derivative, artether and artemether. Of course, these derivatives also in the present market serving the different kinds of malaria. And another compound, Taxol, which is isolated from the bark of Taxus brevifolia and named in 1967. So mitotic inhibitory activity of Taxol has been discovered in 1967. Initially, it was being commercialized to Bristol Myers Squibb and named as Faclitaxel, and Faclitaxel is now approved for different cancers starting from lung cancer, ovarian cancer, breast cancer, and of course different cancers in different years. As Taxol is very scarcely available in the plant, of course this other compound 10-deacetyl becatine is the highly available, highly high yielding compound in the extract. So they used to, the Taxol synthesis, they used to tag the synthetic source to the 10-deacetyl becatine. So this is the synthetic source, synthetic pathway, synthetic process of the Taxol. And vincristine, vinblastine as anti-cancer drugs. These two well-known alkaloids, vincristine and vinblastine, isolated from Catharanthus roseus. These two alkaloids also derived from the based on the bioassayer approach from the based on the traditional knowledge, and these are widely known as a chemotherapy, and FDA approval has been given in 1963 for vincristine, 1965 in vinblastine. So far, these two drugs are the well utilized in the particular even till date, and this is the only source. There is no synthetic source for these drugs. Even Cipla is the monopoly for supplying of these drugs, and also there is a lot of opportunity for the process development of these drugs, as 1 gram of vincristine and vinblastine cost about 6 to 8 lakhs. So this Catharanthus roseus also widely used in the treatment of diabetes in various system of medicine, and based on this discovery, several other drugs also been developed by adding the some of the groups like fluorine group and amide group, and first semisynthetic analog of vinblastine entered in the market called the Vindacin, and Vinrolbin also been developed by adding the indolin group, and similarly by adding the fluorine group, CF3 group, developed vinfluorine. These three derivatives also developed as a drugs and also been approved by the FDA for different types of cancers. Similarly, camptothecin is also one of the best drug derived from the natural products isolated from Campothecia acuminata, and phodophyllotoxin isolated from Phodophyllum peltatum in 1880, and the structure was confirmed in 1950. Based on this phodophyllum toxin discovery, two drugs, etoposide and tenopside, has been developed by adding the two sugar units and as well as two sugars plus thiophene moiety. And these two important anti-cancer drugs was derived from phodophyllotoxin and approved in 1996 by FDA for the cancer treatments. With another just quality control aspects of herbal drugs, as we know, this is the recent, I think 1 week or 10 days back, there was a news clipping which has herbal supplement toxicity lands a patient in the hospital. So these are all happens to the because of the lack of the standardization, because the effect or therapeutic effect or the quality therapeutic effect of the medicinal plants or formulation is due to the presence of the biological active chemicals, which are mostly secondary metabolites, as I told you, like alkaloids, glycosides, saponins, coumarins, and of course 75 to 80% of the medicinal plants required or traditional medicine are supplied from the wild source, not from the cultivated source. So sometimes it may be difficult for the supply because those are all seasonal based. In that case, the vendors are using the other plants which are synonymous to the main plant, so that because of this adulteration, sometimes the quality may be affected and also toxicity also may be developed. So And also the medicinal plants which are traditional used in the traditional medicines are cross pollinated, and sometimes other genetic factors like morphological and genetic factors like a season, and also in temperature and oxygen is also affects these plants to preparation of their secondary metabolites. For example, if you work on the plant, if you would study the plant from south side and Himalayan region, the complete profile is fingerprinting profile is different, because in the Himalayas there is low temperatures and less oxygen, and in the south high temperatures and oxygen. So that makes difference in the plant preparing the secondary metabolites in it. So similarly, lack of consistency in the quality one batch to one batch for the production of the products. Likewise, in the allopathic medicine, like for example, paracetamol 650, either Dolo or other also, if you purchase this type of 650 mg paracetamol, so irrespective of the company, dosage is almost same, but it's not happening in the herbal drugs. So one batch to one batch, they are not maintaining the consistency of the active compound. So that is also biggest problem, quality control problem in the herbal drugs, and also lack of well defined and well orchestrated SOPs. So standardization of the herbal medicines, there are several parameters has to be standardized, like a quantity, dosage, quality, adulteration, and efficacy. These are the major points has to be checked in the standardization of the herbal medicine: physical, chemical, biological, and botanical. In the physical parameter, moisture value, ash value, and extractive value has to be checked. And then chemical value, chemical parameters, the raw material should be free of pesticidal residue and free of heavy metal residue. And in the biological, it should be free of microbial contamination, and pharmacological evaluation, toxicological evaluation should be done. In the botanical side, as I told you, some of the vendors are utilizing the plants which are synonymous to the original plants. In that case, the efficacy is affecting a lot. So in that case, microscopic studies of that plant particularly has to be studied by using SEM and other powder studies. So these are the different analytical techniques which are well approved by the USDA and US FDA, HPLC, HPTLC, UPLC, and LCMS and GCMS. Every technique has its own significance in the characterizing the compounds and standardization of the herbal medicines. So as we are working in this area, just I gave one example. Sasuriya Lappa, which is a very well-known plant utilized in the many of the Indian systems of medicine as a formulation. So we have taken this plant and quantified these two markers, which is a carcinoid and dihydrocosta's lactone, from the raw material by developing a HPLC method. And further, we have also purchased some of the market drugs, which is where this plant is a part of those drugs, and checked by using our developed method whether these two markers are present in that or not. So if it is present, then those are the genuine formulations, but fortunately these two markers are in the detectable level. Further, we also studied the stability studies, where for example, if the active compound is not stable enough in the our intestine, it may be transferred to other compounds should be non-toxic, then only we will survive, otherwise there is a huge effect, as our intestine contains huge pH, acidic pH, like almost to 2.5 to 2. So in that intestine, it has to be stable. So for that, we have also studied the degradation behavior and stability studies. Fortunately, these two markers are stable enough in the stress conditions. So I would like to give some of the examples of marine organisms. As you know, our India has almost more than 9,500 kilometers of coastal region, which is flooded with many of the diverse marine flora and fauna. Here we can see four different types of seas. One is sea is surrounded by the hill, one is flat sea, and sea in between, sea backwater in between two hills. So this also matters for the diversity of the marine flora. And generally, majority of the marine products have been isolated from sponges, tunicates, and echinoderms, bryozoans, a wide variety of marine organisms in their tissues. So these are the pictorial representation of the source of marine sponges, like sponges, molluscas, tunicates, and bryozoa. So an example, there is a one sea animal called sea cucumber. It travels only 100 meters in a day. So while traveling, when it is surrounded by the other big animals to attack on the sea animal, instantly that will prepare some kind of spray. toxic spray to protect from itself by the other animals. So in that spray, in that instant, it will prepare. That is nothing but the defense defense pathway. It will become the novel chemical entities. That's what always the marine source is the richest source, as well as the source of the majority of the potential drugs which has been originated by the originated by defense pathway signal. And traditional sources, terrestrial plants and animals. And see, high level biodiversity in the marine world. High level biodiversity means high chemical diversity, which finally leads to the variety of molecules which show unique structural features and exhibits various range of biological activity. As I told you, in the sea, there is a lot of animals. Of course, there is a competition for survival of each and every animal, and environment pressure, which leads to the biodiversity. And biodiversity means every animal will protect from other animals by preparing some type of chemicals in it, that is called attack signaling, which finally leads to the chemical diversity, and which will finally become a potential new drug. So this is the first drug, gluconotide, which has been derived from the snail Conus magus. It's the first drug of marine origin, which is obtained FDA approval in 2004. And so far, it is also approved for the treatment of chronic pain as a morphine replacement therapy, which is the most powerful painkiller known to date. And this is another drug, Holicandrin B, which is isolated from marine sponge, Holicandria wakadiga in 1985. From this Holicandrin B, as it's showing moderate activity, another drug has been developed by breaking the ester bond, which is called eribulin, which is the most potent drug for the breast cancer, and it is also approved FDA and released in the market, available in the market in 2010. And this is the only drug so far working in the picogram level, and also mostly costly drug. Almost this injection is $5,000. And another compound, bryostatin, which is isolated from Bryogen bugula neritina, and this is also approved in phase 1 studies. Tumor response have been observed, and also approved as a single drug, and also approved as a combination therapy for different cancers. And these are another type of other drugs, anti-cancer drugs, antifungal candidates, and antiviral drugs, which has been developed from the different marine sources. And the drugs from the animals. Several bioactive agents were developed from the body parts and metabolic produce and venoms and toxins of animals. Teprotide is extracted from the venom of the Brazilian viper, and this is also shown at acetylcholinesterase inhibitor, silogepril, and antihypertensive drug captopril. And another epibatin drug also been discovered from the frog, which is a 10 times more potent than the morphine, led to the development of several novel class of painkillers. So there is another kind of natural sources lichens, which are nothing but the symbiotic organism of fungi and cyanobacteria. Lichens also known as a pollution indicators, where it grown non-polluted areas and moisture areas. Lichen we can see when in Sunday, when you buy the biryani leaf material in the packet in the supermarket, in addition to the dalchini, lavanga and biryani, if you will also find lichen, which is a majority of the lichens are edible lichens containing the polyphenolic compounds, specialized compounds. Those are called dipside and dipsidones, and majority of the lichens possessing antioxidant and and diabetic activities also. And utilization of the lichens also known from the ancient times, like plants, and they also having a different various bioactivities. And in India, almost north and south region, particularly Western Ghats and Eastern Ghats, are the sources of lichen. And we also working in this area, lichen chemistry. In the Eastern Ghats, particularly in the Rayalaseema region, we collected and isolated several compounds, and which are shown very good antioxidant activity and other activities. When you see the lichen richest state, Tamil Nadu is a major state which is showing high number of lichens, and followed by Uttarakhand and other states. And another natural source is seaweeds. These are all also called marine macroalgae, and these are all plant-like organisms that are generally live attached to rock or their hard substance in the coastal areas. As I told you, India has more than 9,500 kilometers of coastal region, and containing a lot of marine flora, which is also includes seaweeds. And these are different three seaweeds: red algae, brown algae, and green algae. And green algae is in many of the countries it is utilized as a uh, salad, which is a very high protein salad. Particularly in the Japan, people are consuming this green algae like a salad. And in the red algae, brown algae, there are a lot of uses. They are also part of in different as a like a bio fertilizers and biostimulants and food biopolymers and growth stimulants and nutraceuticals, cosmetics, industrial chemicals. They are also part of the nowadays. Even government of India also giving encouragements, a lot of funds to the state government to encourage the fishermen to cultivate the seaweeds, because seaweeds are a lot of there is a lot of demand in the market, particularly seaweed cultivation. Um there are different types of seaweed cultivation methods: tube net method, raft culture, monolin culture. And in Tamil Nadu, particularly majority of the seaweed industries are there. They also have a lot of requirement of different seaweeds: brown seaweeds and red seaweeds. And agar agar is one of the best product derived from the seaweeds. Similarly, sodium alginate is also one of the best product derived from the seaweeds. And this is a different production of seaweeds in the different states, coastal states of West Bengal, Odisha, AP, and Tamil Nadu, Kerala, and Maharashtra and Gujarat. So these are different applications of seaweeds, like basic utilities, industrial uses, ecosystem services, and even in ice creams also seaweed is utilized on the industrial scale also for production of biopolymers and carrageenan biofertilizers. Seaweeds are well utilized. And when coming to our own activities, as I told you, in the folklore medicine, we regularly interact with the folklore medicinal people or herbal healers to understand their knowledge for the serving the surrounded people based on the folklore medicine. And when we interacting, we will really surprised about their knowledge. And when coming to lab, we will go to the literature first, whether those plants have been scientifically validated or not. If not validated, we will take that challenge to isolate or the active compound or to isolate the active fraction. And then based on that active compound, chemical modification or simplification and derivatives will be developed based on the scaffold. And finally, lead optimization and SCR studies will be studied to identify the active pharmacophore in that compound, derivatives, compound derivatives. And these are the targets we are working: anti-cancer, antifungal, anti-diabetic, and in agriculture side, mosquitocidal, insecticidal properties we are working. And in the applied side, we are actively involved in the standardization of the herbal drugs or multi-herbal formulations based on the marker compounds, and process development of the active phytochemical ingredients, which is not like doing the classical column chromatography. Like it should be industrial viable process. In that case, we have succeeded for the process development of two active compound. One is subbonin and one other is triterpene, and patenting is in the process. And this is a drug discovery process in our CSIR IACT, starting from extraction to drug development. And this is our own working flow. As we are working in different types of natural products, we are also looking after the repurposing of the natural products drugs by screening, as well as by doing the derivatization of the high yielding phytochemicals. So from this, I conclude here. I would like to thank once again to the organizers for giving me the opportunity to share my views regarding the natural products, and also thank my director for his support and different funding agencies. And these are the pioneers of natural product chemistry in India, starting from J.L. Simonson, Ramnath Chopra, Shilamuddin Siddiqui, T.R. Sheshadri, K. Venkatraman, who won the Nobel Prize, and T.R. Govindachari, Asima Chatterjee. She is the first woman scientist who got the Bhatnagar Prize in India, and Susank Chandra Bhattacharya and Sukhdev. He's called a living legend in the natural products. He is celebrating 100 years birthday this year. He is the father of glucurosterones. Similarly, T.R. Govindachari is also called father of neem, as he and his entire life he worked on the isolation of the compounds from the neem plant. And similarly, Asima Chatterjee is also worked on the alkalides. And I'm happy to share that two of my patents were commercialized to Sami Labs, Bangalore, and for further development, after the development, that has been marketed in the international market in the name of Sabroxy tablets, for the nootropics department, nootropics, and these are the major compounds present in that formulation. And we also working on the functional foods where traditional medicinal food plants. So we have developed a two process for the anemia and malnutrition and anti-diabetic activity. We developed a product with the technology has been marketed to Amaya Life Sciences, and they also made a product out of it called Superfoods, which is in the present market for the diabetic. And this product has been launched in IACT March 2023 by the Honorable Science and Technology Minister Dr. Jitendra Singh, Government of India. So finally, I would like to thank for your patient listening, and once again thanks to the organizers for giving me the opportunity. Thank you once again.
Which has nurtured a lot, but there are many areas we need to be. In fact, you have so many talk on these drugs from nature. Uh, the earlier speaker from IICT, Dr. Saab, also highlighted on different perspective on that. So I'd like to give you some idea on that. That is say like how best the perspectives of bioeconomy can be developed using our knowledge with the developing drugs from nature and drugs from our ancestors, that is the traditional medicine. In fact, presently I am working with the Institute of Bioresource and Sustainable Development, which is under Department of Biotechnology, Government of India, and our major target is on this bioeconomy development from the bioresources, obviously with special reference to ENIA Northeastern region when we are working here. Main vision of the institute is to bioresource development and their sustainable use through biotechnological intervention for the socio-economic growth of the region. We have our office at Imphal in Manipur, then another one at Shillong in Meghalaya, at Gangtok in Sikkim, and also in Aizawl in Mizoram. Now, presently, I am talking from Shillong, and we have different perspectives on development of these bioresources of the institute. Coming to the main aspects, like when you talk about the bioresource and developing bioeconomy, where I believe that this pharmacy, specifically the subjects what are pharmacy people we are getting knowledge of, that has a great value on developing the whole bioeconomy of the country, because we are expert of making easy to use formulation for the people. At the same time, we know how to use a bioresource for therapeutic purpose, because that is one of the major goal when we want to put together the health mission. And when you talk about the Indian context, you see that it is a country where we have 15 agroclimatic zones with more than 47,000 plant species. 17 to 18,000 of that is medicinal plant. So such a diverse field area is there from where you have the possibility of developing drugs, developing functional food, developing medicinally potent, therapeutically potent formulation which can be enrooted to any of the regulatory profile that Government of India have. And if you consider the biogeography of our country, no, this is very, very unique. See, we are so fortunate to live in a country where we have the coldest place like the Nubra Valley with minus 57 degrees centigrade temperature in and the dry, cold and desert of Ladakh. We have the rainforest with world's highest rainfall in Cherrapunji, which is 1.5 hours drive from the present place from where I am talking now, from Upper Shillong. And again, we have the evergreen humid tropics of the Western Ghat and the arid and semi-arid conditions of the peninsular India. We have the dry desert conditions of Rajasthan and also the mangroves of Sundarban in West Bengal. So unique biodiversity we have throughout the country. And again, if you think the uniqueness of our tradition, say you travel every 100 kilometers, you will see the language or dialect is differing. You travel every 500 kilometers, you see that we are having a different food habit and completely different uh, cultural aspects, like the traditional food, traditional culture, everything is different. So such a diversity when the country have, and we rarely any country in the world is having such a things. And that's why development of the bioresource, obviously with all aspects of this therapeutic and uh, the other context of developing drugs or pharmaceuticals, that need to be considered, and there are enough scopes for development of these bioresources for bioeconomy. And then why it comes suddenly on that? Say, I hope you, many of you know about this One Health concept, and this One Health concept is one of the major concept. What I want to highlight through this staff development program, even in other places also when we are talking about, uh, as I told that even though I came up with a other institute, but I believe that this is one of the area where our pharmacy friends, they should explore more. And One Health is an integrated, unifying approach to balance and optimize the health of people, animals, and the environment. It is particularly important to prevent, predict, detect, and respond to global health threats, including this COVID-19 pandemic, what we have all witnessed. No? So, this approach can mobilize multiple sectors, discipline, and multiple communities at varying level of society to work together. In fact, these particular aspects, all of us have witnessed during the COVID time, how the varying level of society is working together. So this One Health is one of the basic things what need to be promoted through our pharmaceutical fraternity at a high extent. This One Health involves the public health, veterinary, and environmental sector as well. And this has linked with lot many things, including one of the major prominent aspect nowadays is the AMR, the antimicrobial resistance. And the One Health approach is particularly relevant for food and water safety, nutrition and control of the the pollution management, and many more. So, friends, this is one of the things what we all need to nurture, and where ethnopharmacology has a great role to play. Actually, this ethnopharmacology and integrative medicine, their history tell the future. And if you consider that any of the perspectives of the development of drugs, starting from aspirin, the small molecule from Salix alba, to Taxol, the very big molecule for anti-cancer drug. Again, say quinine for malaria, and again nowadays the artemisinin for malaria. Once again, you see everywhere the answer has come for those deadly disease from plant only, and that too not only plant, the traditional use of those plant. So this ethnic use of those plants and their therapeutic translation, that has a great impact on the whole societal development, as well as the whole context of the drug development what we are thinking about. Now, whether the medicine is specific to area or specific to a group, those are nowadays immaterial because of those concept of One Health. You might have observed during this, again I'm talking about this COVID, because it has given us lot many lessons together, that say people who are using the Ayurvedic kadai or lot of infusion. I believe that no one is here today who have joined in this today's discussion in this program. That no one is there that who has not drank that infusion during COVID time? Everybody. Do we have there those clinical trial of those? No, answer is no. Do we had the uh, any sort of specific evidence that somebody has get cure of COVID earlier? That's why you're taking? Answer is no. And why we didn't? Because we have faith, believe in our system. And moreover, that faith and belief is not only the aspects by which we have to use the traditional thing. By law, it is like that if any drug or any herbs or any system has a 15 years of use which can be predicted by clinical evidence, that can be taken into in the drug. And that is why we are very fortunate to live in a country where we have different ways of promoting this traditional medicine. We have the hydropharmaceutical route, which is very recent. We have the Ayush medicine drug development route. We have the Drugs and Cosmetics Act, by which you can develop a single molecule with clinical trial can go. And also we have that dietary supplement and nutraceutical route, FASSI route. So in any context of that, when you are trying to promote ethnopharmacology, is very important, and it involves wide range of scientists from varying specialties, where pharmacist is one of the major component, I believe. And in fact, good to see that in many of the context of the government perspectives, including Ayush Ministry and other things also, lot many of our pharmacy people are getting absorbed on that particular context. Now, if you go with this one health mission and this ethnopharmacological perspective, you see that there are a lot of aspects starting from AMR, infectious disease, or zoonotic disease, cancer, metabolic disorders. Everywhere we have the perspectives of this traditional medicine, and it has evolved of its own. Say earlier we are using the intact plant, then the extract, fraction, their metabolomic study, characterization of the component, molecular basis of the identification, and then with that in vitro, in vivo study, going to the bioassay guided fractionation, and ultimately leading to the development of the drugs, which can be promoted of any of the route what I told, and then that can definitely globalize the traditional knowledge. And obviously, we feel proud about our knowledge on Ayurveda, where Ayurveda has already told many years ago the Rig Veda about the One Health concept, which was with many perspectives on Charak Samhita, Sushruta Samhita, Bhagavata Nigantha. So, friends, whether you consider the environmental approach, where there is a Briksha Ayurveda, that Ayurvedic concept is there. You think about One Health concept or the veterinary approach, we have that Gau Ayurveda, that is the Asava Ayurveda on horses, or Mriga Ayurveda for the animals. And you think about the human health approach, we have this Ayurvedic Siddhanta, Ayurvedic Niganta, Samhitas. So that is the beauty of our whole traditional knowledge, and it is the beauty of our I should say the whole country that on which we feel proud of, that we have all the system in existence, and that way promoting the things is no a very big thing on that particular context. So when we talk about these particular aspects, no, that there is another sector which we need to give much more importance, and things are developing to a high extent in our country on that. We are very happy, where uh, these traditional health practices or the knowledge empowerment and exchange with the traditional health practitioner is very much needed. You know that we have this Lok Shastra Parampara, where generation together the people are treating the local communities, and those communities together help in development of drugs and pharmaceuticals. If we go on that particular perspectives, no, that this local health tradition, LHT, has a great role to play. And that is coming through the Lok Shastra Parampara, where uh, we are trying to develop a platform for knowledge exchange, empowerment, documentations of the knowledge of these traditional healers. We, with our institutes, even last 30 years of my career, I have been working in this area. So what I feel that if you think about the development of this aspirin, you see that things came from these traditional healers in different way. from the anthropological perspectives. You think about Taxol, again came by the same, and 2015 Nobel Prize came for the Chinese scientist who again work on this, the medicine from the street vendor, where we got artemisinin. So let us respect our heritage on that Lok Shastra Parampara, where India is very pioneer on that. And what we need to do, we need to do lot of innovation and science validation where tradition meets innovation. And in that context, there are a lot many steps, and I think you are well aware about those things where we need documentation, we need validation, and also we need to explore the uniqueness of those traditional practice. This is more important. Say like sometime we used to, yeah, I have seen lot many scientists, and nowadays when we come with the different other fraternity beside our pharmacy people other, we have seen that many people always think about that you get a plant extract fraction, get a molecule is not working, we say that this traditional practice it is not working, but that is completely wrong idea. Say if you believe in these particular practices, and those people are treating by generation together with thousands of people of that village, where 40% of our population depends on this traditional medicine, where those medicine men, they are treating many of the people, and if we are not able to prove science behind it, that is the fault of us, the scientists, not those healers who are practicing them for years together. And here is the point. Then how to prove? Proving is not that to isolate a molecule. It is that we should go work more on the synergy and the combination synergy, that perspectives where it is not single molecule, but molecules together works. And those molecules together, that is a big science. And that need the validation. And we really feel proud that these our ancestors' medicines were documented and validated by that way. And we have these many routes, what I already told, that to develop as a nutraceutical, develop as a phytopharmaceutical, develop as a Ayush drug. And that way we can definitely evolve from tradition to innovation, those knowledge. Friends, when we talk about those things, that major context come into effect is the validation part. And this validation, as I say, we need to concentrate more on the synergy, where this beside the extraction or bioassay guided fractionations, we need to work more on the chemical profile, herb drug interaction, metabolomic study, phytomarker analysis, and the synergy or the combination synergy. Again, if you look at back at our traditional heritage of Ayurveda, that this synergic concept is very much documented there. And many people in the country, many laboratories there also have started working on that. And we have a synergy forum of the European Union, which was developed in University of Bonn, where India, we have a chapter, and we are leading that forum. We'll have a very recently, we'll have a meeting of that synergy forum in India, we are planning. And in fact, I'll tell you something of those at the end of my talk. But mainly, these particular aspects of evidence-based validation of traditional medicine, these are the translational aspects. And nowadays, we talk more about the translational research. And my pharmacy friends, we have lot of aspects in this area. Say you study the herb drug interaction, you study the herb interaction, and you study many more, and then come up with a knowledge on this synergy, combination synergy, network pharmacology through the metabolomic study, which relates with the mechanistic establishment of the whole drug or product, which we have been nurturing from our tradition. So there are a lot of translational component on that, where you do with isolation, then completely with the phytochemical isolation, where we isolate a novel natural product compound which has a effect like Taxol. Yes, it is a multi-billion-dollar business, but all plant, all everything will lead to a single molecule which will work. That we need not to, and we need not to be worried for that. That means that when you are not getting a molecule which works, that means the whole those aspects of the traditional practice is wrong. Rather, we are wrong, we are not able to prove it. So for that, we have to go with the synergy directed fractionation. And this synergy directed fractionation, where we test the fraction of the isolated compound for synergistic interaction and identification of synergistically interacting natural products, which will help in developing the mechanistic approach of the disease orientation. And another one is the metabolomic directed approach. Obviously, nowadays you know that we have all these hyphenated technologies like this LCMS, MS, or GCMS. And with those techniques, you can identify the metabolites present in any extracts or natural products. And from there, from the library of the markers, if you go through, then we can prove the potential leads for the original plant extract or fraction. This, based on that prediction, go with in vivo study and preclinical evaluation, and that way we can go with the metabolomic directed approach, where which is one of another translational component or strategies for validation and evaluation of traditional medicine. Friends, we work mainly on this phytopharmaceutical mission program, where I believe there are a lot many things to do using our traditional knowledge, starting with the documenting them, validating them, and then based on that evidence-based approach, we go with system biology and then prove the synergistic component with this quality evaluation and standardizations of those by extracts or bioactive fraction. And when we develop all the things, there are two major social responsibilities which we must keep into mind, and that has a great impact. Say like we should go with the traditional healthcare practitioners capacity building, their training awareness, and whole the context if you develop, there is a great role on the benefit sharing with the traditional healers. And this benefit means like for our institute, when we are developing a product, the major royalty goes to them, more than 80%, that we have made a rule. We made a publication, their name is there. We make a patent, they are there, one of the holder. So whole the concept of this benefit sharing with the traditional health practitioner is one of the major thing which need to be promoted. I hope this will be included in future in the syllabus of pharmacy, where the next generations, the students should know that how best we can respect our own tradition or heritage, which being possessed by our forefathers uh, for a long, long time, and how we can develop drugs from there. That is why I used to call them that these are the drugs from ancestors and drugs from nature. So if they were not using those ancestors, knowledge was not there. So never ever we should have these traditional practices so far. So now how this synergy can work, I'll just tell you some example. Many of you might be working on this particular context. Just take the example of this Lagnaria sirica, Loki. You know, this is one of the very well used vegetables for hypolipidemia, uh, uh, hyperlipidemia people are using. But in many cases, you'll see that these hard drug interaction studies are not being done. So this is one of the major contexts what we need to. And here, if you say when we go with this with LCMSMS profiles and network pharmacology for the prediction of the molecular mechanism, we show these extracts contain a lot of metabolites on which when we interpret it on the synergistic interaction with the in vivo studies with animal, that correlate well, and by that way this combined in vivo and network pharmacology analysis relates with the relevant targets on hyperlipidemia. And this hyperlipidemic use of that particular medicinal plants, in fact, has a great role to play in establishing the structural features of that. And when we talk about those particular perspectives, this again lead with many, many contexts, like say, last before year when many of us, we are working on this that which plant or plant product having use under coronavirus. So there is a plant Andrographis paniculata. I do not have that slide here now, but those Andrographis paniculata has a high impact on the society, on this this cytokine, and we have been able to prove that mechanistically that how does andrograph is manipulated, if why it is effective so against the COVID coronavirus. So this is one of the context on which many of our scientists nowadays are working, and I want to highlight here more on that you think on this how best we can establish the multi-molecular mechanism of action instead of the single molecular mechanism of action, because this is one of the context which is very much needed in our own perspectives for development the product process technologies, which not only give uh, faith or our own system or re-established our own research, but also promote the whole traditional knowledge scenario to explore drugs from nature and drugs from our ancestors. So, friends, the major context of my talk today, what I want to tell is that how best we can go into the translation based on our tradition. And this tradition to translation, this relates with so many contexts, starting from your field study to extraction, fractionation, standardization, making a formulation, again standardization of the formulation, developing the final products in the clinical trial, and leading to the clinical trial which establish the therapeutic validation perspectives, then develop them through a uh, product with different route like Ayush route or FSSI route or FSI route or phytopharmaceutical mode or whatever it may be. So this translation doesn't relates only to development of the product, but translation relates with our own work on those particular perspectives where we can establish the mechanism of actions of many of our products. And this really is one of the hindrance, I should say, in promoting our own product. Say, I have faith on my own traditional medicines as because from childhood, we are being using those things. But a Western world person may not have that much of faith on our system until and unless we have scientific evidences. So to develop those scientific evidences based on the traditional use is one of the major responsibilities of the researchers nowadays, and where again pharmacy students researchers has a great role to play, and in fact that should be one of the major context of the translational component of the research from traditional medicines on which we have been working with. And I would like to emphasize here that when you work on exploring this traditional medicine, these explorations means therapeutic validation, chemical validation, and mechanistic evaluations of the use of the traditional medicine. Remember one thing, that here we are not doing something rocket science, but we are doing much more of the social aspects, because if you can re-establish the fact that why this plant X is used for memory enhancer, if you can establish that one, then that is one thing which will relate many of those people who have less faith and believe on the on this particular system, not only in India but abroad, and that too to promote our own product globally. So there are enough scope in this area when we work on the translational component of the traditional medicine, and there are enough scope for development of patent. And IPR is one of the major context in those translational component, where you developed any of those contexts when you search uh, in the system, you will see that very less work has been done on that. I'll tell you some interesting story here which may give you interest to work on that. Say, when we talk about research in this area, I have seen being editor and editorial board of around dozen of journal. What I have seen that we saw anybody and everybody want to highlight on writing a review article. And uh, why? You review for your own. Don't think that you will be making an article when in the area which you have not worked with. Just think about something on those translational component based on your review, rather, that how best you can translate the mechanism of actions of the drug or the therapeutic validational aspects or the chemical validation aspects. And uh, we have no limitation of what you say instruments or other things in Indian laboratories now. There are enough instrumental facilities, and many of those national laboratories, they have a facility called Sahaj. For say, if you do not have, you may not have LCMSMS at your institute, which is very costly, but you can avail that facility using that Sahaj portal of any of the institutes, national laboratories, including us. And in that portal, you can the minimal payment, and you can do your work. It is not always that uh, instrument is not that, that's what we are not able to. It is like an intention that matters a lot rather. And I'd like to emphasize that let you take those particular aspects, how best we can work more on the translational component on that. And to highlight this particular concept, no, that uh, myself and my I work with Professor Wagner's. Many of you know the Wagner's reagent, alkali test reagent. He was my guide at Munich, and also Professor Robert Varport, on which I worked for a long time. And this is one of the concept things. What it was a concept paper. Later on, we have developed that one as a book, and I'm so glad to get their patronage, which Elsevier has come up with the document on quality control and evaluation of herbal drugs, where because when we work in this area, what the inhibition, what I have felt, that we do not have a complete guide in this area. We have to go with hundreds of documents, and to get the idea and how we'll do bioassay guided fractionation, how we'll do LCMS study, how we'll go with the combination synergy or safety study, or even the in vitro and in vivo evaluations of the drug. So this is a context on which we highlights on the traditional system, and to harmonize them, starting from ethnopharmacology, ethnomedicinal perspectives, quality evaluations of the herbal medicines, then qualitative studies, morphological, microscopical evaluations, starting with and leading to extraction and other downstream processing for herbal drugs. Mind it that this is a very big area, and nowadays this downstream processing is getting more important because we have so many industries who are producing herbal medicine, but very less raw material supplier. And so this technology for extraction need to be, and this downstream processing has a great role now. So, and then lead to this bioactive component, and making different analytical parameters for that, for the quality evaluation study, leading to thin layer chromatography, high performance thin layer chromatography, high performance liquid chromatography, then liquid chromatography coupled with mass spectrometry, leading to chemical profiling and the marker analysis for the quality control and evaluation of herbal drugs. These all perspectives had been described in this document, and with many context with live examples, and also on the bioassay guided vaccination, like leading with this enzyme inhibition studies or evaluation of herbal drugs for antimicrobial, parasiticidal, antiviral study, and finally the metabolomic study, which is the need of the hours, leading with the synergy establishment with safety related quality issues for development of herbal drugs, and also quality assurance and the stability testing. So if if this is available, and also we have come up a very recent document. This is not in our slide, on the evaluation and validation of herbal medicinal products. This is also another context. Friends, when we talk about those all translational component, at the end we feel proud about this our Atmanirbhar Bharat vision. We're encouraging the startup program under this, and if you come up with all those context of translational research, there are enough scope in this area to with the involvement of the rural and local communities, could lead to knowledge empowerment and exchange, where we're learning through drugs and from nature, or from our ancestors, could truly lead to innovation from tradition. Yes, we need a lot of scientific validations, and that is where our major role is. Again, through those startup, we can have ethno entrepreneurship. This is one of the major things which we have taken up with our institute and many other institutes, where recognitions for researchers and community people working in the field, that is very much needed. say traditional healers who has lot many things in his hand. They should have this serving the community. Their capacity building training is very much required, and at the end, developing industrial linkage, which is more important. So this ethno entrepreneurship is one of the context where I would always suggest my students and even many of the faculties who have their interest say like we can develop a sustainable societal bioeconomy incubation model with ethno entrepreneurship, and this is very much needed to explore the drugs from nature and drugs from our ancestors. And we know that there are a lot of platform on that. BIRAC's DVT is there, Department of Biotechnology BIRAC program is there, and many other programs are there where they promote on this entrepreneurship. Let us take this entrepreneurship on the which there on this from the bioresources specific traditional knowledge, where and which can help in developing a platform for knowledge exchange and empowerment with benefit sharing of the knowledge holder, and encourage the startups to promote this Atmanirbhar Bharat, specifically through this development of nutraceutical, phytopharmaceutical, or even with the herbal extracts, where the traditional knowledge inspired drug development can be promoted for the bioresources, for the commercializations and the benefit sharing. So this is one of the aspects what all the pharmacy institutes should think of, because this is one of the, it is not that we will always be a job seeker, rather we should be a job giver. We should give job, and that is a great satisfaction, and this is possible. And presently government of India has taken a lot of step on that, where through this Atmanirbhar Bharat Abhiyan you have enough scope for promoting this particular context. So promote, encourage, I would like to encourage all of you and our students through you that rather promote your entrepreneurship skill and give a instead of being a job seeker, be a job giver, and that is very much possible through this traditional knowledge validations and model through this ethno entrepreneurship. And on this particular context, in fact, when we talk about this for drug development, procurement for our traditional knowledge or this bioeconomy incubation model, this bioeconomy development is very much possible from when you explore the knowledge from the ancestors. Friends, on that context, we took a lot of initiatives, and one of the initiative was the establishment of the Society for Ethnopharmacology India. In fact, you might be, all of you know that our former president, no, Dr. APJ Abdul Kalam, he was very much well-wisher of this particular area. And in the Rashtrapati Bhavan part of that, developed the herbal garden. And from then onwards, I had a link with him, and I was blessed to get his many advice. One of that was to establish the Society for Ethnopharmacology India, which is a society constituted by eminent academicians, researchers, industrialists, with a vision for providing environment for knowledge sharing among the researchers, healthcare practitioners, decision makers who are interested in ethnopharmacology and natural product research. And this society, which was made under his mentorship in 2012 and 13, it got the whole affiliation with the International Society for Ethnopharmacology, where it bridges the gap between academia and industry and act as a resource for the students, researchers, traditional healers, policymakers, industrialists, and those who believe in natural remedies. So you can go through the website of the society, www.ethnopharmacology.in, join in that. This was, I'm so happy that again with the encouragement and support from Dr. APJ Abdul Kalam in 2012 when it was inaugurated, no, that Nobel laureate Luc Montagnier, he came and to India. It was in Calcutta, Science City, and the Indian chapter was evolved. And now we have 17 local chapters throughout the country, and all the local chapters starting from Jammu to Jorhat. Everywhere the chapters are there. So you can link with this we were involved with this founding, and now this is being this society is affiliated with the International Society of Ethnopharmacology, and in fact, being the president of this International Society of Ethnopharmacology last 2 years when I was, so we promoted many of those Indian contexts. Last year we had the 10th year celebration of that society, where we got very huge gathering of again national and international pharmacologists, ethnobotanists, phytochemists, anthropologists, and all to promote this particular area. So you can join that society and explore the opportunities uh, to explore specifically this particular area where the use of medicinal plants in traditional societies, and seek to understand the cultural and pharmacological dimension on the human use of medicinal plants that can be explored in a very, very different way. So this is one of the contexts we can explore. So with this, I stop here, and I believe that these bioresources are the nature's gift for everyone. Let us promote and preserve those gifts. And in fact, with all our involvement together, definitely we will be able to do more in this particular area for developing drugs from nature and drugs from our ancestors, which will not only give us more proud to feel that we are promoting our own system, but this will pave a way for promoting our own things globally. And that is why through our institute, and even what I was telling, the Society for Ethnopharmacology, we took the major role that globalizing local knowledge and localizing global technologies for promoting the bioresources, which can lead to development of bioeconomy uh, at large. Thank you once again. Namaskar, Jai Hind.